With the number of COVID cases at more than 300 million around the world, effective vaccine is a key to stopping this global pandemic. And currently, messenger RNA seems to offer the best hope. Even though more than 800 million doses of messenger RNA vaccine have been administered and proving to be efficacious, many people still don't understand the fascinating history behind the vaccine. Welcome to Prince Mahidol Award PMAC Interview. I'm Pavit Pimvichit, and joining me at the Mandarin Oriental Hotel today are the recipients of the 2021 PMAC Award who will talk to us about history, the present, and the future of the mRNA technology. Drew Wiseman is an immunologist and a professor of medicine at the University of Pennsylvania. Catherine Carrico is a biochemist, a professor at the University of Szeged, Hungary, and an adjunct professor at the University of Pennsylvania. She is also currently the Senior Vice President of BioNTech. And Peter Collis, a professor of biochemistry and molecular biology at the University of British Columbia in Canada, and the Director of Nanomedicine's research group, UBC. Okay, so I think we start off with Dr. Wiseman. Can you tell us what is the function of a vaccine?
So vaccines have been around for 400 plus years. What they do is that they make an immune response against a pathogen. And the idea is that that immune response in your body prevents that pathogen from infecting you or causing disease. So it's a way of stimulating protection against any pathogens in the vaccine that you might encounter at some point in life.
Okay, so how many types of vaccines do you have currently? Other than COVID?
Yeah, there's many. There's about 20 that are commonly given to children every year. And if you talk to a parent, they'll complain bitterly because it's every 3 months, every 6 months, they're going to the pediatrician who's sticking needles in their arms. So it's about 20 vaccines that are given 3, 4, 5 times for each.
Yeah. How many types in terms of like, is it a live attenuated, is it a dead vaccine, dead virus?
So there's many different platforms as well. The first ones were live attenuated, vaccinia. There are then inactivated, like influenza. There are protein subunit, like tetanus toxoid. And the newer vaccines are the adenovirus, the viral vector, RNA and DNA.
Okay, so to you, Professor Carrico, back in the 1990s, the concept of messenger RNA is not very popular in the scientific community, I think, and you were working on that. And why was it not popular?
In the 1990s, it was more popular to work with DNA as the gene therapy program started actually in 1990, and as new genes were discovered, everybody tried to implement some kind of program to replace or supplement, but it is in a DNA base. And the RNA was known very well that it is very labile, and both in shelf life-wise, so that it couldn't stored quite a long time, and also when it delivered to the cell or in the body, it very quickly degraded. So when we started to work with RNA, of course, it was always a problem that the protein was produced for a very short time and the level was not sufficient. So we had to work a lot to make the RNA last longer, and had to improve the RNA structure that so more protein is produced so we could detect, and it was eventually became physiologically relevant.
Okay, before that you were trying to use messenger RNA to cure diseases, and then you switched to making vaccine when you met Professor Wiseman. What did he say to you?
Yes, so my goal was always to make mRNA coding for a therapeutic protein, as I work in cardiology and neurosurgery and try to treat those kind of disease related to those organs. And when I met Drew at the Xerox machine in the hallway in the Department of Medicine, really I was there, and then he mentioned that he's interested in vaccines, and I mentioned that I can make mRNA, and that's how we started to work together.
Okay, if you had internet back then, you would have met?
Probably not.
Okay, what are the theoretical advantages of messenger RNA that's better than other types of vaccine for COVID?
So, we're just beginning to understand that. And if you look at all of the COVID vaccines made so far, and the FDA and EMSA and other organizations agree, the RNA is the best. It gives the highest responses. There are a couple of mechanisms for that are complicated science, but it turns out that RNA LNPs give phenomenal antibody responses that give you protection against variants, which is the most important thing nowadays.
Okay. Well, the native mRNA is not stable, as you mentioned earlier. What did you two do to make it work?
So one of the problems was not just it is not stable, but it was that it was very inflammatory. And although at the beginning we thought that maybe that immunogenicity is good for a vaccine, but turned out that it was not. But as I was mostly interested to make mRNA coding for therapeutic protein, so this inflammatory response was a no-no. And so we start to understand why it is immunogenic, what is this inflammation is coming from, what is sensing. At that time it was not known. And then we tested out different RNA, and we discovered that if we modify the nucleosides, the RNA will be not inflammatory. And that would be feasible using in vivo, in animals and others. And Drew was doing more research in vaccine direction and discovered that this type of RNA is also better for vaccine.
Okay, and how did you get the mRNA for the spike of the coronavirus?
So, the spike protein of coronavirus was known for 30 years to be the important antigen, the important gene to put in a vaccine. That goes back to even before SARS and MERS. The unknown was how to stabilize it. So that's work that Barney Graham worked on, which is putting the antigen in a form that makes a protective response. So what Moderna and Pfizer did is they used a stabilized version of spike, and it worked well. I actually made a vaccine in Thailand with the Chulalongkorn VRC. We used an unstabilized version. It actually works better. So it really comes down to being an unknown. It's trial and error, finding the best way to make a spike or any other protein for a vaccine.
Okay. Other than the COVID mRNA vaccine, is there any other development of a mRNA vaccine for other diseases?
There are so many, I probably don't have enough time to list them. They include diseases like malaria, TB, influenza, norovirus. They include diseases like cancer. They include things like food allergies, peanut allergies, and treatments for autoimmune diseases. So it's expanding every year. So the number of potential diseases for vaccines keeps increasing. And then on top of that are all of the different therapeutics that can be developed with RNA. It really is an unbelievably vast platform of potential treatments with RNA.
Okay, so now we come to you, Professor Cullis. Don't forget, we didn't forget you. Anyhow, so their work is basically the payload, and yours delivery system, the LNP, the lipid nanoparticle. Tell us a little bit about that.
Okay, there's a long history here. It goes back almost 50 years. I was originally a physicist, and then I switched into chemistry, and I was really investigating the properties of biological membranes, and particularly the lipids. There's a lipid bilayer in there that's quite vital to life. And in other words, it provides the envelope that cells use to separate the inside from the outside. Anyway, so I got fascinated by the properties of these lipids, and in order to study, there must be, thousands of different lipids in any biological membrane. So it's very hard to study it all by itself. You have to make what's termed model membranes. They're very simplified versions of a biological membrane. So we made these model membranes, and then we realized we could use them as drug delivery systems. We found ways of loading cancer drugs, for example, inside these little vesicles, these little nanoparticles. And then so that's where I got interested in drug delivery. I mean, can we get cancer drugs more specifically to where they're needed? Because right now they go all the way through your body, and that's not a good thing. You know, your hair falls out, you throw up, and all of the nasty side effects that you get. So we worked in that area for about, about 10 or 15 years. And then the challenge of gene therapy came along. So we thought, okay, well, maybe if we can deliver these smaller molecules, we can do something with the bigger ones. Now, the problem that we hit was that in order to get these negatively charged molecules associated with the lipid nanoparticles, you need positively charged lipids, and they're very toxic. And so we came up with an alternative way of getting a positively charged lipid that at physiological pH values, the pH in your body was relatively neutral and non-toxic. And that was where we got very lucky, because it turned out that this is a way of encapsulating RNA and DNA. But it was also a good way of delivering things inside a cell, as we found out, that in addition to being able to encapsulate, we could also use, if they got taken up in a cell, then they would break out of the little compartment that they're in when they're taken up. They would break out and get in and deliver the contents to the cytoplasm, and that was really luck. We're not really expecting that. Then, of course, so in the course of this, I'd started up a small company. We were contacted by Drew and by Katie to say, okay, these things are delivering. We had them so they could deliver messenger RNA to the liver, and then the protein would be produced. We weren't thinking about vaccines at all. But then the suggestion was that maybe we should test these as vaccines, because if they're producing a protein, maybe they can produce a protein that's associated with a virus and produce an immune response. And again, there was a bit of luck here, because it turned out that they worked very well as vaccines. It just points out, science is not a very linear path. You don't go straight; you go all over the place, and then things, interesting things happen. And it's certainly the case.
So how do you control where the LNPs, which cell is going to?
Well, you have to take advantage of biology. What these lipid nanoparticles, they will adsorb. They associate with certain proteins that are already in the body, and once they associate with those proteins, that triggers uptake into cells that are in the surrounding area. So these are things that many times you can't prophesy what's going to happen. You have to do the experiment, and then you say, "Wow, that's interesting." In the case of the vaccine, it proved particularly interesting. They actually worked very well.
Can you imagine any other way to deliver the mRNA into the cells other than LNP?
We feel very lucky to have got one way.
Okay.
There are potentially other ways, but they tend to be kind of mechanical. You're banging stuff in, and so much more invasive. The cells are not very happy with those sorts of mechanical treatments.
Okay, so how did all your discoveries come together into a COVID vaccine? How did that come about?
Yeah, the first was Katie and I meeting over the copy machine,
right.
And we kept talking and we kept working together, and then it was time to find a way to deliver the RNA. So we knew about lipid nanoparticles, and we contacted Peter and Acuitas. We also studied about 40 other formulations and found that the lipid nanoparticles worked the best, and that's how it came together.
Okay. Well, can you summarize the efficacy of a mRNA vaccine for COVID at this time?
So that, if you compare the efficacy to other things, like other vaccines in the disease, the vaccines give you 5 times higher levels of antibody than getting infected with the disease. The vaccines give you 3 to 5 times higher levels than any other vaccine platform. They also give you other forms of immunity, cellular immunity, that gives you protection. So a respiratory virus with 90+% efficacy was unheard of before COVID-19. We were used to influenza that averaged 50% and could be as low as 19%.
Okay, well, as parents of the mRNA vaccine, how do you reassure us of the safety in terms of LNP and the messenger RNA technology?
I think you just have to look at the world. Close to 1 billion people have been immunized with RNA vaccines. They are incredibly safe. There have been a couple of very minor adverse reactions, a couple per million, that are minor, that are easily treated and go away. There's been nothing else. There is no severe unknown adverse events. It just hasn't appeared. After 1 billion doses, it isn't going to appear.
Okay. Are you satisfied with the speed and the rollout of your product?
Well, I think none of us are ever satisfied with anything, which is why we're researchers. I mean, you know about the LNP rollout.
Well, yeah, I mean, just with regard to that question, though, I mean, from the sequencing the SARS-CoV-2 virus to getting a vaccine actually in the clinic was 3 months. I mean, how much faster than that can one get? And then there was the 8 months where the clinical trial was going on. So this has to be the fastest that any vaccine has ever been developed before, going from scratch. With regard to the LNP, there are advantages there in terms of manufacturing because it's not a cell-based process. You can get all the raw ingredients and put them together in a very, in a very straightforward manner, and so that allows, from a manufacturing point of view, allows one to scale up quite remarkably. We also take advantage, of course, as Drew was saying. I mean, these things are very, very potent. And so we have incredibly small amounts of messenger RNA in there, 30 micrograms for the Pfizer-BioNTech vaccine, for example. So a teaspoon of messenger RNA will treat 1 million people. It's spectacularly potent.
Fascinating. And any countries can manufacture this?
Well, that's the big question. So I've been working with Thailand for the past year. And with Chulalongkorn University, we made our own mRNA LNP vaccine. And then we outfitted a GMP production site. So that's specialized facilities for making drugs and vaccines. So right now, they're making drug that's in phase 3 trials and will be distributed to the Thai and 7 surrounding countries in the near future. What's important about that is when COVID is over, they'll be able to make any vaccine they want. They can make dengue vaccines, which is still a big problem in this region. They can make HIV vaccines. They can make whatever vaccine they want.
Okay, great. Well, let's talk about the future. How are you going to update the vaccine when there's variants coming out quite regularly, quite fast?
I mean, you have to see first, because whether you need a new vaccine for the new variants, because as we learn from the new variants for the SARS-CoV-2, that the original Wuhan vaccine, which was generated based on that sequence, was as protective or was a little bit less protective. So, by the time you would roll out a new clinical trial, then a new variance is coming. So we have to see that the variance which is emerging is now is such that the original vaccine is not protective. Then it can be made, the new vaccine, and as you already know, that it takes about 3 months to deliver. So if we learn the sequence.
Okay, well, another problem that we face here is the cold chain to keeping the vaccine during delivery and also the shelf life. Are we expecting any improvement in these fronts?
Yes, of course. Exactly how that's going to happen, we're not sure. But one of the things you have to remember is that there was a very short developmental time frame here between when the vaccine was, the need for the vaccine was discovered and then getting into clinical trials. Normally, pharmaceutical companies would spend a year or more working on the formulation to get it stable and and have the right shelf life, etc. Well, of course, they really didn't have time to do that, and so they ended up with formulations or lipid nanoparticles containing mRNA that were, they needed special treatment, such as a cold chain treatment. But there's no inherent reason why that needs to be the case, and I suspect we're going to see 4-degree, refrigerator systems or stable systems in the near future. It's just it's really a matter of adding the right excipients, as they term it, to these formulations, and you can get them pretty stable.
That will be wonderful, I think, for working in the fields.
Mm-hmm.
Also, what do you see as the further step with this technology in terms of mRNA and also LNP? What diseases are we going to be curing in the future?
Well, Drew's already indicated quite a few. The interesting thing right now is, in the liver, there's all sorts of opportunity, and you can, in addition to potentially curing liver diseases, you can use the liver, as we term a bioreactor, to make proteins that might do something else elsewhere in the body. Make monoclonal antibodies that will attack a cancer, for example. But the part that's fascinating me at the moment is, can we get these things to other places in the body following intravenous injection? And so, for example, if we can get to bone marrow, then sickle cell anemia and various leukemias, we can see ways of tackling those. Get things to the lung, and cystic fibrosis and many other lung-oriented diseases. Get to the brain, and boy, that's where it gets really interesting. Mm. There's all kinds of opportunities there for curing brain diseases. So it's an extremely exciting time. It's a revolution in medicine that's going on right now.
Well, we published last week that we can make CAR T cells in the body. So instead of having to take cells out of a patient and expand them and give them back, we injected RNA LNPs and made CAR T cells in a mouse and cured their disease. So it opens up a future of many different therapies where you give a single injection of RNA LNPs for gene therapy, for cancer, for autoimmunity, for just the number of diseases is enormous.
Sounds amazing. Sounds like a wonderful future.
For quite a long time, the most advanced therapeutic application of mRNA was to delivering in the heart, injecting to the heart during bypass surgeries. And this was actually went on phase 2, and now it's advancing phase 3. So there were already application of mRNA for other diseases. It was just, the vaccine and the problem, the pandemic pushed the mRNA in the front line.
Excellent. And I heard that you told your surgeon colleague that he will be obsolete in the future. So it seems like your words are coming true. Okay.
Oh, I was just, exaggerating things, but
I hope you're still friends with him.
Yeah.
Anyway, well, thank you so much for coming on this interview, and I hope we get to talk to you again soon.
Thank you.
Thank you.
And you're watching the PMAC interview series. Stay tuned and hope you subscribe to our YouTube channel.